Hello, everyone, and welcome to today's live session on getting started in 3D models. This is a live Q&A session to enable attendees to ask questions about growing and characterizing 3D cellular models. Today's seminar is brought to you by LabRoots and sponsored by Thermo Fisher Scientific. For more information on growing 3D models or any educational resources on 3D models, please visit www.thermofisher.com/3D models. My name is Robert Horton, and I'm a Senior Product Manager at Thermo Fisher Scientific, and I'll be your moderator for today's event. With us today, we have a panel of scientists from Thermo Fisher Scientific that routinely grow a range of different spheroids and organoids models. We have Dr. Mark Kennedy. Mark is our lead R&D scientist with our 3D model program and is directly involved in the development of new cell culture products for the growth and characterization of 3D models. And also, we have Dr. Richard Josephson. Richard is the lead scientist in our neurobiology group at Thermo Fisher Scientific and is primarily focused on developing neuronal 3D models for the purposes of studying neurological diseases such as Parkinson's and other neurological disorders. As mentioned, our goal today is to provide a forum for participants to ask questions about growing and characterizing 3D models. We encourage you to participate by submitting as many questions as you want. To submit a question, just simply type the question in the box that says Ask a Question box, and click on the send button. We'll answer as many questions as we have time for. If you have any trouble seeing or hearing the presentation or the questions, click on the support tab found on the top right of the presentation window, or report your program by clicking on the Ask box located on the far left of your screen. With that, let's get started. I first want to hand it off to Dr. Mark Kennedy for a quick introduction on 3D models to help stimulate some questions. Mark.
Thank you, Robert. And thanks everyone for joining. So as Robert mentioned, while folks are busy entering their questions that they would like us to try and answer into the Ask a Question box, I'm going to take just a few minutes here to go over just a couple of slides, just some very high-level background on 3D models to sort of help us get started and introduce the topic a little bit. So, in the next couple of slides, all I'm going to briefly go over, one is to just sort of highlight what, reinforce what Robert said is the sort of the objective or the goal of this Q&A webinar session. And then we'll go through just a couple of slides on the background and an intro to 3D cell cultures. We'll spend a couple of minutes, maybe the bulk of the time, talking about the sort of the key ingredients in making 3D models in general. In cell culture type workflows, and then we will turn it over to the question session, and we'll do our best to answer as many questions as we can. So as Robert mentioned, really the point of this webinar is to just create an open forum, a live event, if you will, where we can interact with customers and people, especially people who are new to working in 3D models or interested in getting started in 3D models, and perhaps we'll be able to answer some questions, maybe provide some guidance, or pass along some lessons learned, which sometimes are hard lessons learned in working in these types of workflows. So, as I'm sure many folks who have joined to listen into this webinar and hopefully participate, some of this type of information is probably not super new news. One of the things I want to highlight here, as I'm sure people are aware, that 3D models are definitely an emerging field in cell biology, particularly in cancer research. Over the last decade or so now, many new methods have been published that detail ways of creating different types of 3D models, so things that are either 3D spheroids that may be just a single cell type that are cultured as a cluster that provides somewhat more in vivo-like cell interactions, to more complex models like organoid or stem cell derived models that sort of are often co-culture models or models made up of different cell types and that are architecturally and functionally similar to an in vivo organ. And so many of these have been made to represent lots of different tissues in the body. including things like liver and brain and intestine and a number of others. And these are also useful for studying different types of disease as well. And as it says here across the top, that it would appear at this point that certainly the application of these organoid models in particular is certainly leading the charge in new avenues of research in the cancer space. Many of these models can be made from different types of cells. As I mentioned, you can make them sometimes from different cancer cell lines that maybe you're used to growing as a 2D monolayer in your lab, or other types of primary cells. Many of these models are also induced pluripotent stem cell-derived models or embryonic stem cell-derived models. But a lot of the models are also derived from adult tissue, particularly resident stem cells in different organs that reside in the epithelial compartment of these organs. Are another source of cells for making these types of models. And so, as I've already sort of mentioned or alluded to, really when we're talking about 3D models, we're talking literally about a 3D assembly or cluster of cells that are obviously much different than the traditional 2D monolayer culture systems that are the prevalent model over many decades in cell biology and biomedical research in general. And the switch to 3D models is really driven by a desire to have cells interact in a way that more mimics how cells interact with each other in an in vivo environment. And this has led, as I mentioned, to the development of many different types of spheroid and organoid model systems. And it's really also being driven by different tissue engineering approaches, things like bioprinting, that are allowing researchers now to generate these more complex assembly of cells for doing various types of research. And sort of straddling both of these sort of broad classes of models are things like traditional co-cultures, where maybe you mix two different cell types that typically are found adjacent to each other in an organ in an actual animal model. And also, we have the tissue-engineered organ-on-a-chip type approaches, or sometimes known as microphysiological systems. These are sometimes, highly engineered 2D model systems in some configurations, and in some other configurations, they may mimic more like an organoid or spheroid type model. But in general, when we talk about 2D models and 3D models, these would be the broad classes of these models and how we think about them. So, a popular question if you're new to this area or something you're just hearing about more recently, a question you may have is, why are researchers moving into these systems? Sort of, what's all the hype about? And, largely this is driven by the desire to make better cell models for studying human biology. And what I mean by that is, traditional 2D monolayer cultures, although are incredibly helpful and have been very informative over a very long period of time and will continue to be so into the future, they are somewhat overly simplistic and have less than natural sort of cell-cell interactions, and this leads to changes in cellular physiology that may not represent what a particular cell behavior actually looks like in an in vivo setting. And so these 3D model systems, some of the interest in driving researchers towards 3D models is to sort of overcome that as a limitation of 2D models and put cells in a context where they can interact with other cells in all dimensions and interact with different cell types in a spatially important, or I guess a spatially important way, where different cell types are arranged similarly to the way in which they might be in vivo, and things like that. And some of the results that have been coming out of this is that when you culture cells in this way, the proteomes and transcriptome signatures of cells in 3D models tend to look more like the signatures that you would find when you look at in vivo cells, whereas it's quite commonly seen that in 2D models, once cells are adapted to a 2D culture system and and growing on plastic, that a lot of these gene and protein expression signatures don't always look like what the in vivo situation looks like. And so, these are some of the features of 3D model systems that are driving people to think of them as more physiologically relevant cell culture models. That's not to say they aren't with their challenges. Of course, many of the existing workflows that researchers are sort of been trained on and are comfortable with are all 2D workflows, and so being able to adapt to culturing cells in 3D formats is certainly it's challenging, and sometimes simply switching from one to another is not overly straightforward. And this is particularly true when you think about the types of assays that are available, and the ability to either lyse a microtissue versus a monolayer of cells, or the ability to label cells that are not on the surface of a spheroid for doing imaging type analyses and things of that nature. So there are certainly some trade-offs and challenges still to be met around workflow compatibility and assays. And of course, this all leads to an increase in complexity, which is good, but that also makes things just in general more challenging to work with. But I think as more and more research is coming out on these models, the growing consensus is probably that it's the added challenges, once you overcome them, are going to be worth it. And so this sort of naturally brings us to this next question, particularly for people who are new to 3D models, is how do you get started? And sort of the short answer to this is it's the same as you get started with any cell culture model system. First of all, you need to identify what cell types you want to work with, what cell types are important to you, and there are considerations then about how to sort of QC your cells. How do you know your cells are useful or going to be helpful for an assay? And given that a lot of 3D workflows tend to be very long and somewhat more reagent intensive, making decisions about quality of cells early on is very important to contributing to the success of your experiments. And so, an example of this is looking at, the pluripotent status if you're working with iPSC cells. If you don't have a good QC assessment of just how good your iPSCs are, then every time you form a 3D model with these and differentiate them into different types of organoids, this is going to contribute to a lot of the variability that people see in sample to sample and experiment to experiment. So, being able to have QC tests that kind of create a standardization, if you will, within a workflow is definitely something that people need to be aware of. Another obviously key ingredient here for culturing these things are the use of sometimes matrices, such as different types of basement membrane extracts, and also specialized culture vessels or culture vessels with specialized surface treatments. So many 3D models, in order to make them sort of either grow in a 3D sort of configuration, and also to help enhance their differentiation and physiological relevance. Sometimes cells are grown sort of embedded or fully encased inside of a basement membrane extract. So instead of growing cells on top of a thin coating, now you've got to deal with this material that's typically temperature sensitive, so you have to work on ice, which limits scalability in many ways. So it's definitely another thing that a researcher needs to be aware of. And then with the plastics, a number of these type of 3D model systems may also require the use of certain specialty plastics. I think many of these today sort of fall broadly in a category of non-adherent type plastics or super low attachment plates, and this allows cells to, or it helps cells or convinces them to interact with each other more so than trying to interact with the surface, because they can no longer bond to the plastic of the surface. So it helps facilitate the formation of 3D models in many different types of systems. And the third piece, of course, are the media and supplements that you need to grow your cells. Choosing good quality media choosing growth factors and small molecules and other types of supplements that are reputable, are consistent from batch to batch, and that sort of thing, is definitely something one needs to pay very close attention to, and sort of I think put a lot of weight behind when you're sourcing materials for growing these types of models. And the last piece, which is sort of invisible and not really shown here, are assays. I mean, it's great if you can grow these, but if you can't assay them and actually, get something useful out of them at the end, then that's a whole other set of questions, I think. But really, what I want to point out here about assays is what I mentioned a couple slides ago. Is there's a lot of assay optimization that still needs to be done to work with 3D models. Being able to label cells that are, more than just the cells that are on the surface of the 3D model is not always a trivial undertaking, especially if you're working with antibodies, if you're working with different types of chemical probes for fluorescent labeling. I think those are a little more manageable right now based on existing protocols, may only need some more minor tweaks. So that's the sort of food for thought that I'd like to leave everyone with, and hopefully some of that spurs some brain juices and some questions that you can submit to us. And with that, this is the panel that Robert introduced earlier. We did have unfortunately a team member who was unable to join us today, but we would like to turn it over to the Q&A, and Richard and Robert and I will do our best to answer any questions that you have.
Great. Thank you, Mark. I really appreciate the really quick summary. Hopefully help stimulate some questions. So as I mentioned, the whole goal of today's session is really to have a live Q&A session to help answer your questions. So if you do have any questions you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll do our best to answer as many questions as we have time for. There's already quite a few that have phoned in, so I'll do my best to give us a little bit of diversity and answer as many of these questions as possible. So with that, let's start out with a really general question for both Mark as well as Richard. When you first got started working in 3D culture, what were some of the challenges that you had to overcome that are just, common practices that you do today? Maybe you could just give us a little insight on that.
Sure, I can go first.
Sure.
So it was a big adjustment initially. A couple of things that I mentioned, or at least one of the things that I mentioned during the preview for some of the models that I first started working with temperature-sensitive basement membrane extracts was sort of a new level of difficulty that wasn't in any of the traditional 2D workflows that I had. Although I use these materials for creating coated plates for growing different types of pluripotent stem cells and those sorts of cells that require that kind of coating. Working with thick, or I should say highly viscous basement membrane extracts was definitely one of the challenges that I was a little bit unprepared for, and there was a little bit of a learning curve there. Another sort of, in hindsight, a very obvious challenge that came up was probably the first time that I formed spheroids in a non-adherent U-bottom plate. I seeded my cells and they formed beautiful spheroids in a day or two, and decided, okay, it's time to change the media. And I take the plate out of the incubator, I slide over to the hood, I turn on the aspirator, I pick up the plate, and I go, I'm going to lose all my samples. So figuring out ways to do a media change on a sample that is not attached to the bottom of a plate without losing your spheroids, that was something that, in a hindsight, is obviously a very important challenge.
Right.
But definitely something that required a little extra effort and sort of standard operating procedure optimization in terms of making that less of a headache while working with these types of models.
Right. Yeah, I'm going to go back a little further than that even. So when I started working in 3D models, I thought it would be easy. Just take my cells growing in 2D, dissociate them, plate them in suspension, and voila. But it was not. We were initially trying to generate spheroids in agitated culture plates. And I had to learn the hard way that You have to set your speed very carefully when you're doing that, and control your media volume so that all your cells don't float to the center of the well and aggregate. And so the solution to that was to start using the low attachment 96 well U-bottom plates, where we found for neural progenitors that you got a very linear response to the number of cells that you seeded per well and the diameter of the spheroid that you would get out. So that made things much more predictable than the mass culture of spheroids in a shaking plate. And then from that U-bottom plate, you can then, if you need to encapsulate, you can easily collect the one spheroid per well into a pipette and encapsulate from there.
Right. Good. You know, both of you guys kind of mentioned a little bit about the challenges of media change, and we actually, and it turns out we have a question about that. Maybe you could expand a little bit more. You know, the question was sometimes we lose our spheroids during media changes. Do you have any recommendations on how to overcome some of that?
Well, what I do is not change 100% of the media at any time. I change the media more often than I would in a 2D culture, but I change only half of it, or some set percentage of it each time, so you're leaving a small volume. And if your spheroids sink to the bottom, that's enough generally to protect you. But I don't aspirate you pipette off that media carefully.
Yeah, there are definitely a couple of I guess, tricks to doing that. I mean, as Richard mentioned, you know, doing a 50 or 75% media change is definitely the way that we went early on when we were working on this problem. And sometimes then when if you're exchanging a whole different media cocktail to another one, you would just do it successively to dilute away any trace amounts of the first media. And, you know, for some of the differentiation experiments, that was probably important. You know, the other things that we've done more recently are to sort of adapt I guess you'd say it's automated equipment, but things that a lot of labs have, like a plate washer that may be used normally for doing ELISAs. We found that, you know, some of the models of these systems have enough programming capabilities that you can safely aspirate to a certain level down into the well that allows you to change media on a full plate. But then the other thing that, you know, the only other thing I would add on top of that is, you know, make sure you do regular, you know, QC. Don't just assume you have all your samples when you get to the end. Mm-hmm. So typically what I do is every day whenever I have a media change day, I literally either inspect every single well, or if you're lucky enough to have a microscope with an automated platform where you can, you know, take a single bright field snapshot of every well, that you can then look at sort of in one picture and identify any samples that are lost, so you can sort of monitor your samples accordingly. But really the only other way, I would say, is to, as Richard mentioned, is to, you know, percent media change.
Mm-hmm.
Yeah. And sticking to the culture theme, and you guys both mentioned media as well. You guys are playing perfectly into the set of questions that are coming in. Congratulations. Maybe you guys can comment a little bit more about the media formulations. You know, are people primarily using, you know, the general medias like RPMI, DMEM? Do you need specialized media? Talk to me a little bit about what medias are used and what kind of media formulation, what achieves really good results.
That's a good question. I mean, so the 3D field is still somewhat new, and kind of what that means to me is that, you know, when you think about the 3D models that are out there, they've all been built on the existing 2D model systems. So frequently, I think you'll find that, you know, the whatever basal media, for example, that was being used to culture a comparable cell in 2D was probably the starting point for anyone who was making the switch to 3D. And in many cases, that's the media, I think, that has stuck. In some model systems, there are, you know, some advanced versions of these media systems, and this is particularly important if you're trying to work in a serum-free environment. So working without FBS, using some of the advanced media formulations is definitely something to consider. And I guess the biggest change really is around the way in which we supplement the media and at least when it comes to stem cell derived models, the amount of supplementation of small molecules and growth factors and that sort of thing that's required in order to culture the cell type of interest. I think those are some of the things that I would point out.
Great. Thank you, Mark. Appreciate it. Switching gears a little bit, because I mean, after you have your model, obviously you'll want to characterize it. So there's obviously a couple questions around what methods have you guys used so far in characterizing your models, and I think this is a question for both Richard and Mark. So basically, if you give a little insight of what assays or characterization you've done in your models so far, and if you have any tips or tricks when starting to use these various characterization methods.
Well, obviously in neuroscience, the gold standard is activity. So we do record from neurospheres on our neural spheroids on a microelectrode array plate. And it can be very, very difficult to get good results with that. To even plate spheroids down, we're finding is a tricky process. So, so there aren't that many people who have successfully recorded from spheroids. You can't really stick a patch clamp in them and record from them. So immunofluorescence is the other major assay. It can be very tricky because neural spheroids are very, very dense to get any information about the interior. So there are protocols for clearing the tissue that work, but they're extremely labor intensive because they go through successive dehydration, rehydration steps. And then you need the right imaging equipment, confocal, to get your data. So a lot of people in the neuroscience field are doing single-cell sequencing or single-cell RNA, which requires you to dissociate the spheroid and recover cells and sequence RNA. But that is tedious and expensive. So, I mean, it's very important to choose your assay early on so that you can optimize for that from the very beginning.
Yeah, and the only other thing I would probably add to that, I mean, we still do some of your, the types of routine qPCR-based assays, right, not necessarily going to sequencing or single-cell sequencing. But, we do have some evidence even that if you're going to lyse a spheroid, and I'm sure the larger the spheroid gets, the more significant this problem is, and it may vary, admittedly, from cell type to cell type, but in some cell types, we do find that results are highly variable if you, use certain types of lysis reagents. I mean, some of the tried, tested, and true methods seem to work best. Using things like Trizol seem to be best, at least in our hands. And sort of in addition to that, when possible, we've sort of taken to a freeze thaw event, which I guess in theory kind of creates a little bit of a freeze fracture effect when you go to use it. This seems to be giving us a better lysis of these spheroids when we want to do that kind of work. But and we've also done other types of metabolic type assays on hepatic models as well, different types of luminescent enzyme or metabolic enzyme activity assays. They generally work. So a lot of the reagents like that are out there can be optimized for assaying these reagents. They just require more effort than the standard 2D version.
Right. And have either of you guys tried any of the plate-based readouts, or have you used your organoids or spheroids in a plate-based reader yet?
Yes, and I know some of our colleagues in a different segment of Thermo Fisher have done a lot of this, working with spheroids in plate reader assays and also in automated high-content analysis platforms. Certainly, this is something you can do. There are considerations like Richard brought up about, there may be a need to look into, more labor-intensive methods such as clearing in order to try and get more signal from within your samples. That said, there's still, depending on your imaging equipment, there are still going to be limitations to how many cell thicknesses you can actually peer into a 3D model without actually going and having samples either cryo-sectioned or sectioned in paraffin blocks and analyzing that method. But of course, that shoots down any type of throughput.
Right.
So unless you're looking at a small number of things, that's the opposite of probably what you're asking. Other plate reader assays where cells are sort of encapsulated in a bed of basement membrane extract, we've done those with standard viability type assays like Presto Blue. They also require optimization around the length of time for incubation with these reagents. Again, this seems to be directly related to permeability of these through the BME and also through a number of cell layers, and then being able to detect that in a plate reader. It certainly does work, but there's as with most assays, there's effort that needs to be put in there to optimize time of reagent treatments and cell seeding density, which is not trivial, to make sure you're well within a good range of being able to get reliable readouts.
Mm-hmm.
Yeah, and just a quick question along those lines. When you guys do have to use an ECM or a basement membrane extract or something like that, do you guys typically feel that you have to remove it in order to characterize the models? Does the ECM interfere with any of the characterization methods that you've experienced, at least?
No. When I've done it with the neural spheroids, I can stain for at least the outer surface of the spheroid through the ECM. I do find that fixing in paraformaldehyde tends to break down that ECM. That's a known issue, that paraformaldehyde will cause it to dissociate. So oftentimes they'll leave the cells stained and left behind, but they'll be somewhat less organized than they originally were.
Yeah, and we've done some of that too. We've in sort of a mid-throughput, working in 96 well format, we've had similar sort of challenges. We typically work with phenol red-free material where possible. I think this seems to help reduce any, sort of background signal that we may get from the ECM. But, as Richard pointed out, when you fix these samples, it definitely depolymerizes, paraformaldehyde will depolymerize some of these things. And so that's a challenge. We found we can get around that by post-fixing with paraformaldehyde and glutaraldehyde. The only challenge with this is this potentially creates high levels of background, so then it becomes a trade-off of sample loss versus higher or poorer signal-to-noise ratios. There are some background suppressor reagents out there that I've tried that seems to help, at least in certain assays. I think the name of the reagent is called something like Backdrop or something like that. And there's also a so-called background suppressor in one of the molecular probes called Cyquant Direct that also seems to do a pretty good job of suppressing that background. So if you are in a system where you don't want to remove cells from the ECM, if it's important that they stay where they are, then that seems to be a trick that seems to be working well for us so far, is to post-fix it with glutaraldehyde and then using this background suppressor.
Great, thank you. A few questions about your opinion questions. So do you think 3D models will ever replace animal models? And what do you think are the current limitations of 3D models to be used to entirely mimic an in vivo system?
That's a big question.
Yeah, it really is.
I know, it's a loaded question. I apologize, but it's a big one.
It's a very good question, a very important question.
I don't know about will they ever replace animal models, but they won't replace animal models anytime soon. And a lot of that has to do with the ADME considerations, administrations of the drug, metabolism of the drug, that depend on many different cell types. And whereas where you can make organoids that contain many different cell types, oftentimes the proportions of those cell types are not well controlled at this time. So, and you need cells from many different tissues. You might need liver tissue in a brain model to really cover what happens inside an animal.
Yeah, studying systemic problems is definitely something that, maybe that's where, organ on a chips, I guess, that approach, may have a future.
Right.
But I think I agree with Robert, with Richard that, I think in time there may be a reduction, but the likelihood of eliminating seems to be a far off thing. Just because, some of these cells will continue to behave, there are still advantages to working with some of these models in vivo. For example, if the effect is somewhere else, a lot of these 3D model systems don't contain any vasculature. And,
right.
and that in and of itself is a limitation to how big and probably how complex current models can actually become. And, so depending on your question of interest and the model system you're working in, that could be a very important limiting feature. But in some models, it may not be.
Right. I mean, that said, the pharma companies are very eager to eliminate animal testing as much as possible.
Very expensive.
So it's easy to imagine a first wave of testing in organoids followed by more focused testing in animals to save costs.
Great. And then, obviously, one of the challenges today, I would argue, is just reproducibly and consistently growing the models, right? And we actually have two great questions along those lines, and I'm going to combine them into one. Yeah. What is the typical percentage of organoids that are successful from the total generated in an experiment? And the second question is very closely related, and they were asking how reproducible is the production of neural organoids starting from iPSCs with today's protocol. So really, they're both asking, what is the current success rate from your experience? What's normal? And any comments you can add there?
I don't know how to answer that, how successful the overall total is. For the neural organoids, so we've been making a lot of neural spheroids starting from neural progenitors, and that is quite reproducible when you start with a tool like the U-bottom plates, where you can get predictable sizes from seeding. But when you're starting from iPSC, it depends on the protocol. So if you're choosing a protocol where you start from embryoid body formation, the sizes of those are not necessarily as controllable, and the percentage of the cells you want within is not as controllable. So that adds quite a bit of noise to the reproducibility, I think. That said, we're working on some products that will make reproducible PSC models much easier to get in 3D in the near future. So.
Yeah, and the only other thing I would add to that, I think, is the consistency and reproducibility is also wrapped up in the which type of neural organoid you make. So, some of the published protocols result in a model that's far more complex in terms of number of cell types that are present, and so as you can imagine, these things start out without any sort of axes, and it's one of the cues that's missing from, say, an embryo, that cells know where to develop and help control proportions of cell types and that sort of thing. So, the more complex system you're trying to make, I think the more challenges you're going to have with reproducibility and consistency.
Right. Yeah, it's a direct trade-off, complexity and reproducibility.
Exactly. And so as you slide down the scale to somewhat less complex, where maybe you're only modeling corticogenesis or something like that, where, while still obviously very complex and lots of cell types, it's still a reduction in cell types. And some of the published models that we've seen, at least on the face of it, they seem to be more reproducible. And, depending on what research question you're asking, level of complexity that you need is probably just an important consideration as well. For that very reason, is that you may struggle to get enough samples that are consistent if you're working with a more complex model, but if you don't need that complex model, you may just get better data with a somewhat more simplified version of that type of an organoid, something that, represents a smaller region of a given tissue.
Right. And I would imagine success rate would vary based upon the model yourself you're looking at, right? Even starting from the cells that you initially used to form the model. Yeah.
Absolutely. Yeah, no, there are definitely lots of things that contribute to it. I'm sure there's variability between, especially with iPSC lines.
Yes.
I'm sure there's variability with embryonic stem cell lines as well. Some of them have propensities to, they seem to differentiate into certain lineages a little better than others. So, if you're looking at sample to sample or experiment to experiment or cell line to cell line, there are different degrees or different, types of reproducibility and consistency concerns that, I think a researcher needs to pay very close attention to make, sort of broader claims around experimental results.
Yeah. And we have a question here about variability within a plate. So just based upon your experience, does the 3D structures vary a lot from well to well within your different cultures that you tried? Again, it may be tissue specific or model specific. Just curious on your experiences.
I mean, at least for me, I don't want to speak for Richard. For me, I mean, it's similar to the previous question, right? Some of that reproducibility in architecture is related to, difficulty or inability to control proportions of cell types. And so, they may not necessarily look exactly the same if you've got varying numbers of cell type A, cell type B, and cell type C.
Right.
So it's hard to answer sort of specifically, but that's what I would say.
Great. All right. Okay, shifting gears a little bit here. Your experiences in using 3D printing to make 3D cell models, and any insights you can share there.
That's a good one. Yeah, so I've not done a lot with bioprinting just yet. It's certainly a growing area of interest. I think to date it seems to reside a little more on the engineering or tissue engineering side of biomedical research. And I guess kind of what I mean by that is it's a lot of the work with bioprinting is probably done from an engineer's perspective, which in and of itself is very interesting, and it's a potentially a great way to control some of the consistency problems where with reasonably good resolution you can control for architecture by printing a certain number of cell types in a specific geography or spatial arrangement, and do that much more reproducibly than some of these other self-organizing models like you get in organoid systems. I would say, it still seems to be a bit of a new field. While there are, people making headway in the space, a lot of it is still very much, I think, on the discovery is the wrong side, but still on the development side. For me, it's still sort of I'm keeping a close eye on it with lots of interest, but not a lot of practical experience in that field yet.
Great. All right. A couple of questions here about what ECMs we have used and been successful with, especially for organoids.
Well, primarily we've used the gel tracks for encapsulation of spheroids or organoids, and had pretty good success with that. It seems to work fairly reproducibly. Mark has tried a few things more than I have.
So yes, mostly it's been in that class of product though. Gel tracks is maybe for obvious reasons is our go-to. But the other comparable products that are on the market, we've basically seen very similar results regardless of which version you're using, at least within the the model systems that we've been exploring. So, that the class of basement membrane extract is, the bulk of where we've done our work. A little bit with collagen as well. That's probably, more well studied, and it's a relatively defined reagent compared to the basement membrane extracts. We've had some success there as well, but largely we've been working with basement membrane extracts with, comparable results across all technologies.
Right. Yeah, we've also had for the neural spheroids when we want to attach them, had good success with laminin, especially for trying to attach them to a multi-electrode array. The laminin is very helpful.
Yeah, and I know I've had this question before, and I see it again now on our list. So for systems that don't use or scaffold free, that use something like a ultra low attachment plate or a hanging drop apparatus, do those spheroids eventually secrete their own ECM, or do you have any insights there?
I think it depends on what type of cells are in the spheroid. There was a presentation neuroscience meeting last year where somebody showed sections of neural organoids they were making that were secreting laminin, and the laminin was present as a halo around the outside of the spheroid. That was very striking.
Mm-hmm. Okay. Good. And then we also have a question about where we obtain our cells for 3D culture. Do we always frequently source from patient biopsies, or where could one go to get cells?
Yeah, so I guess I can add a little bit here. There are a number of different sources that one can go to for cells for different types of 3D models. There are a number of different cancer cell lines, sort of the workhorses in many labs. Quite a number of them have been shown in recent years to, be able to self-assemble into some sort of organ or a spheroid type culture that behaves differently than its 2D counterpart does. So that's one source of cells. There are different types of primary cells that people have been using as well. I think there's a growing popularity in the use of using primary human hepatocytes and animal-derived hepatocytes as well. So these being the adult hepatocytes, these are non-proliferative cells. And while there is batch-to-batch or isolation-to-isolation, donor-to-donor variability, companies are qualifying these cells to sort of take that burden away from customers at least. To show that you can culture these in a round bottom or hanging drop or one of these types of formats, and form primary cell spheroids as well. And that's a cell type that's obviously very important in drug discovery. Otherwise, iPSCs are very commonly used. So kind of wherever you get your iPSCs or whether you derive them yourself, I'm sure there is some variability in the efficacy. We've seen a little bit of that between some of the embryonic stem cells that I've worked with in recent years. But in general, they work. Sometimes there are tricks that you can apply to help convince them, if you will, to aggregate a little more efficiently. Sometimes the use of things like ROCK inhibitor or Vitacell in certain cell types is something that may help there. And of course, there's a very significant population of publications now that have demonstrated, the ability to isolate cells from, or progenitor and stem cells from primary tissue, whether animal-derived or human tissue-derived, either, in resections or patient biopsies. And this can be either normal cells, or they can be cancer tumor cells, tumor-derived cells, where you can basically, grow pieces of the tumor ex vivo in a way that is very analogous to making PDX models in mice, only the benefit is you don't need any mice. So really there's a wide range of cell types or cell sources that people can look into for their cell of interest. And there are quite a number of I think public biobanking efforts underway to help tackle that and to become a resource or a repository for researchers who may not have access to patient tissue and things like that to be able to source these cells as well.
Great, thank you. All right. A question around your experiences with kidney organoid cultures, and if you ran into any problems or challenges when growing kidney organoids.
I haven't done that.
I've done iPSC-derived kidney organoids once in the past. You know, I forget how many. There are a few of similar but different published protocols out there. The ones that I've tried actually worked pretty well. I think, I forget which lab did the original publication off the top of my head. You know, these are tricky in a sense because they're probably one of the more complex organs in terms of number of cell types that would normally exist. I don't know how, I'm not sure how many of those cell types people have been able to generate in a meaningful way. But, you know, the published protocols generating, you know, proximal tubule cells, collecting duct cells, you know, basically a good part of that piece of the kidney. The protocols were relatively reagent light compared to many other organoid models that I've worked with. And, you know, they work reasonably well.
Great. Okay. And then I'm going to combine two different questions here together. A couple of questions around genome editing of your organoid models as well as gene silencing, and just curious on your experiences in editing cells that are eventually used for 3D models, or even editing the 3D models themselves, or if you have any guidance or advice in that arena.
Yeah, I don't think that editing the 3D models themselves is that common an approach, because you end up editing the cells on the outside and not the inside. But it is very common to generate edited iPSC lines and then form organoids from that. I'm currently doing organoids from some edited lines, iPSC lines, and there seems to be a good deal of reproducibility from line to line doing that. So, I mean, I think that starting from the initial iPSC line is probably the way to go.
Yeah, and that's the approach that I've seen taken as well, to generate sort of disease-relevant mutations, or to make isogenic lines, whether they're iPSC lines from patients, or, you know, to make other disease or normal controls. And also in the adult stem cell field, you know, there are growing reports on being able to do that. But as Richard said, I think doing the gene editing in the singularized or near singularized cells before you grow the organoid is going to be the most effective approach. Otherwise, you're going to, as Richard said, you're going to end up with cells. You're going to end up with a mosaic, basically, where only the outer cells or some percentage of the outer cells are the ones being gene edited. And, you know, given that these models may have multiple cell types, they may not even be the cell type that you want edited. So if you were to do it, you know, starting at the 3D stage, you may not be setting yourself up for success. Definitely start with the starting population.
Great. And I think we only have time for one last question, so I'll ask it to both of you, both Mark and Richard. Same question to you guys. If you had one tip for anyone who's brand new starting in 3D culture today, what would it be? It's a hard one, I know.
My tip is not a very, well, the tip that I would give, I guess, based on my experience is plan.
Right.
You know, these protocols tend to be pretty long. They're, you know, for a lot of people, if you've been working in a lab for a while, but working with, I guess, what today we would call more traditional models, and you're trying to make the switch to a 3D model, then, you know, there is a little bit of a learning curve on these things, on, you know, being able to adopt these culture systems. You know, it's a different way of looking at cells. The analysis, you know, is more challenging. So planning ahead, thinking about, you know, the amount of reagents, because these do tend to be more reagent intensive as well. So thinking about the amount of reagents of certain growth factors, you know, given that you see lot-to-lot variability sometimes in these types of reagents, or if you need to prepare conditioned media. You know, I tend to think that, you know, spending more time on the prep work, on the planning, is what I would recommend to people, and sort of, you know, the second piece to that is, I think there's a saying I heard some time ago that, you know, 6 months in the library, or a week in the library can save you 6 months in the lab. So this type of planning ahead and making sure, you know, you're not just doing something because you can, but, you know,
Right.
applying that level of focus and forethought is, when possible at least, is definitely what I would say to people.
Right. I think you also need to think hard about how you're going to maintain the viability of your cells, because it can be much more challenging with diffusion limits. Keep a spheroid alive or an organoid alive. So you need to figure out how you're going to try to keep them small, keep them well fed, which may require more frequent feeding than you're used to in 2D. So it can be quite labor intensive over a long period of time.
Great. Excellent. Well, thank you both. You know, we're out of time, so I'll have to end the conversation there. But before we go, I'd like to thank Dr. Kennedy and Dr. Josephson for their time today, as well as the audience for joining us today in their long list of questions. We did not get to all the questions that were submitted. So if you do have any questions that were unanswered, we will be reaching out to you, or, you know, feel free to reach out to your Thermo Fisher representative to help answer that. So, so in addition to thanking Mark and Richard, also want to thank LabRoots and our sponsor Thermo Fisher Scientific for the webcast today. And the webcast can be viewed on demand. LabRoots will alert you when via email when it's available for replay. And of course, we encourage you to share this email with your colleagues or anyone who has missed the event today. So thank you very much for attending, and until next time, thank you and goodbye.